Mitochondria is the powerhouse of the cell. Now, it is called this because mitochondria are responsible for generating the majority of the cell's energy in the form of adenosine triphosphate, or ATP. Mitochondrial dysfunction involves the deterioration in the structure and function of these cellular organelles. Now, quite interestingly, the prevailing theory of how these organelles came about is known as the endosymbiotic theory. It is hypothesized that mitochondria are the descendants of free living bacteria that were engulfed by primitive eukaryotic cells. Over time, this engulfed bacterium developed a mutually beneficial relationship, with the bacterium providing the host cell with energy through aerobic respiration, whilst the host cell provided the mitochondria with protection and nutrients. Support for this theory comes from the fact that mitochondria have their own DNA, distinct from the nuclear DNA of the host cell. Both mitochondrial DNA and bacterial DNA are found in circular form, unlike the linear DNA found in eukaryotic cells. It also lacks the histone proteins associated with eukaryotic nuclear DNA. It is relatively small in size. Human mitochondrial DNA averages about 16,569 base pairs, whilst human nuclear DNA is over 3 billion base pairs. Mitochondrial DNA does not contain introns and has its own ribosomes for protein synthesis. In this process, it uses what's known as bidirectional replication, a process that, you guessed it, is also used in bacterial DNA. Mitochondria also shares similarities with bacteria in terms of their size, shape, and replication process. Additionally, the inner membrane of mitochondria, where many of the important metabolic reactions take place, bears a striking resemblance to the cell membranes of certain types of bacterium. Mitochondria play a central role in energy production through oxidative phosphorylation. They convert nutrients and oxygen into ATP, which fuels various cellular processes. Before entering the mitochondria, glucose is first converted into pyruvate through a process called glycolysis, which occurs in the cytoplasm. Pyruvate is then transported into the mitochondria. Pyruvate is further processed in the mitochondria through the citric acid cycle, sometimes referred to as the Krebs cycle. During this cycle, pyruvate is oxidized to produce carbon dioxide and high-energy electron carriers, including NADH and FADH2. These stand for nicotinamide adenine dinucleotide and flavin adenine dinucleotide, and are both coenzymes that play crucial roles in the electron transport chain during respiration. The NADH and FADH2 molecules generated in the citric acid cycle enter the electron transport chain, sometimes referred to as the ETC, which is located in the inner mitochondrial membrane. The ETC consists of a series of protein complexes. As electrons pass through these complexes, they move from higher to lower energy states. This flow of electron releases energy, which is used to actively transport protons, or H+ ions, across the inner mitochondrial membrane from the matrix, the inner compartment, to the intermembrane space, the outer compartment. The transport of protons across the inner mitochondrial membrane creates a concentration gradient, with a higher concentration of protons in the intermembrane space and a lower concentration in the mitochondrial matrix. This gradient creates an electrochemical potential, and the potential energy is harnessed to produce ATP. ATP Synphase, also known as complex 5, is a protein complex embedded in the inner mitochondrial membrane. It acts as a molecular machine that uses the proton gradient to generate ATP. As protons flow back into the mitochondrial matrix through ATP synphase, they release energy that is used to convert adenosine diphosphate and an inorganic phosphate into ATP through a process known as chemiosmotic coupling. Mitochondria are both a source and a target of oxidative stress. The electron transport chain, which is essential for ATP production, generates reactive oxygen species known as ROS as a byproduct. Over time, This can lead to damage to the mitochondrial DNA, proteins, and lipids, impairing mitochondrial function. ROS can cause mutations, leading to errors in DNA replication and repair mechanisms. Oxidative stress can also cause proteins to become misfolded or otherwise malfunction. In addition, it can lead to cellular senescence and the release of pro-inflammatory factors, contributing to chronic inflammation, another hallmark of aging. Unlike nuclear DNA, mitochondrial DNA is more vulnerable to damage because it lacks the protective mechanisms present in the cell nucleus, such as histones, leading to a tenfold increase in mutagenesis. The creation of new mitochondria is through a process known as mitochondrial biogenesis, and this can decline with age. Reduced biogenesis leads to a decrease in the number of functional mitochondria, impacting overall energy production. Mitophagy is the selective removal of damaged or dysfunctional mitochondria by the cell. Impaired mitophagy results in the retention of dysfunctional mitochondria, which increases the production of ROSs and exacerbates damage. As these mitochondrial functions decline, cells experience energy deficits, affecting their ability to perform essential functions. This leads to a decrease in tissue and organ functionality over time. Mitochondrial dysfunction is associated with several age-related diseases, including neurodegenerative disorders such as Parkinson's and Alzheimer's disease, cardiovascular diseases, and metabolic conditions such as type 2 diabetes. Strategies to preserve or restore mitochondrial health are areas of active research with the potential to promote healthy aging and improve overall well-being. One of the drugs of research is known as J147, which was first developed in 2011 and entered phase 1 human clinical trials in February 2019. The drug initially showed the possibility to reduce Alzheimer's in mice. Recently, however, research has shown that this J147 drug works by manipulating ATP. In its presence, neurons were protected from the aging-associated toxicities caused by dysfunctional mitochondria and standard ATP production. It does seem to be that the reduction in these reactive oxygen species is a key step in reducing the aging process. Dave Sherbert, the head of Salk's Cellular Neurobiology Laboratory, had this to say, and it is a quote I fully support. People have always thought that you need separate drugs for Alzheimer's, Parkinson's, and stroke, but it may be that by targeting aging, we can treat or slow down many pathological conditions that are old age associated.